Proteins by Caius Van Hout,
John Hughes,
and Liam Sweeney.
What is a protein? A protein is a large biological molecule made of amino acids, and it plays essential roles in the body, including structure, transport, enzymes, and signaling. Every protein has a unique shape and function, and that function is determined by the order of its amino acids. Proteins are produced through the central dogma of molecular biology, which describes how genetic information flows from DNA to RNA to protein. In the nucleus, DNA holds the instructions for building proteins. When a cell needs a protein, a specific gene on the DNA is copied into messenger RNA through a process called transcription. This mRNA acts like a temporary copy of the gene. Next, the mRNA leaves the nucleus and travels to a ribosome for translation, when protein synthesis occurs. The ribosome reads the mRNA in groups of 3 bases called codons, and each codon corresponds to a specific amino acid. Amino acids are organic molecules that consist of a central alpha carbon atom attached to a carboxyl group, an amino group, a hydrogen atom, and a special R group that determines the property of the amino acid. These R groups are responsible for the 20 standard unique amino acids found in nature. Transfer RNA molecules then bring the correct amino acids to the ribosome, matching their anticodons to the mRNA codons. As each amino acid is brought in, they are joined together to another amino acid through peptide bonds, in which a carboxyl group of one amino acid joins to an amino group of another acid, releasing H2O as a byproduct. This is the primary structure of a protein, the linear sequence of amino acids. Remember that the type of amino acids and the pattern that they are lined up in determine the function of the protein.
The secondary structure of a protein refers to small repeated folding within a polypeptide chain. These structures are formed through hydrogen bonding between the carbonyl oxygen and hydrogen of the backbone. Unlike the primary structure, the secondary structure does not rely on the R groups. Instead, they have 2 major secondary structures, the alpha helix and the beta pleated sheets. In an alpha helix, the carbonyl oxygen of one amino acid forms a hydrogen bond with the other hydrogen from an amino group. This creates a stable coil. Beta sheets, on the other hand, consist of extended strands of polypeptides aligned side by side. These are also held together by hydrogen bonds, and the strands run anti-parallel in their most stable form. Together, these help form the protein shape. The tertiary structure of a protein describes its 3-dimensional shape. This shape is a result of the R groups of amino acids that are far apart in primary sequence but brought together as the protein folds. This level of folding determines nearly all aspects of protein function. Several forces and molecules contribute to tertiary structure. Hydrophobic interactions are key in the formation of the tertiary structure, as the nonpolar side chains containing carbons and oxygens move away from the water and refuse to mix with it. As a result, they cluster up and cause the chain to collapse, giving it its shape. Disulfide bonds form between 2 sulfhydryl groups, and their bonds are very strong compared to other forces, as they are commonly found in proteins living in extreme environments. Hydrogen bonding and van der Waals forces are weak attractions within an atom that each rely on electron configuration in their own way that help give the protein its shape. While most proteins get the right shape, prions are infectious proteins that lack both DNA and RNA as they are misfolded. They primarily affect the tertiary structure, as the sheets are different from that of a healthy protein. Healthy proteins have a 3D structure made up of primarily alpha helixes, which are very coiled and flexible. However, prions are rich in beta sheets. When bonded, they become rigid and stack side by side, exposing amino acids to the surface, allowing them to react and change their function. These prions are very dangerous, as they can infect and cause the misfolding of other proteins. When introduced to other proteins, these prions change the protein's shape to theirs and clumps up with it, killing neurons in the process. This is the reason for many diseases such as mad cow disease and Jacob's disease.
The structure a protein goes through is the quaternary structure, which is the part of the protein that exists when 2 or more folded polypeptide chains, called subunits, are assembled into a functional complex. These subunits can be identical, or homomers, or different, which are heteromers, and their arrangement gives rise to properties not possible in a single-chain proteins. Quaternary structure is stabilized by many of the same interactions seen in tertiary structure, such as hydrophobic interactions, hydrogen bonds, ionic interactions, and sometimes intermolecular disulfide bonds, but now acting between subunits rather than within one. A classic example is hemoglobin, a tetramer consisting of 2 alpha and 2 beta globulin subunits. Hemoglobin exhibits cooperative binding, meaning the binding of oxygen to one subunit increases the affinity of the remaining subunits, which is an emergent property that only exists because of the quaternary structure. Quaternary structures allow for regulation, enhanced stability, structural flexibility, and the formation of larger molecules machines such as ribosomes, which consist of dozens of proteins and RNA molecules. There are many types of proteins that are in the human body as well, one of those being enzymes. The primary function of the enzyme is that they act as catalysts, which are meaning that they speed up the chemical reactions that go on inside the human body by binding to specific substrates. And a substrate is simply a molecule that attaches to an enzyme in order to initiate a response. One example of this phenomenon is the substrate glucose and the enzyme amylase. Once the substrate binds to amylase, this creates a hydrolysis reaction that breaks down the starch into simpler sugars, such as glucose and fructose, which is important for cellular respiration. A common way to remember an enzyme is that all the known enzymes today end in ase, such as lactase and carboxylase. The suffix ase originally came from the term diastase, which was the first enzyme ever discovered in eggs.
Thank you
For
Watching.